Thank you. I think the microphones need to be a little bit lower for me. Great. So thank you very much to Bo and Natalia and NNI for inviting me. So I'm going to talk a little bit about a large study that we were involved in running, looking at lactoferrin in preterm infants. I've got a few disclosures. I've had funding from government sources, but also charities. But most importantly, my main disclaimer is that I'm a clinician and I'm not a basic scientist. And the talk today is going to cover some aspects of preterm infant nutrition in general, some of the challenges we face, but particularly focusing on NEC and sepsis. So most of what I'm talking about is relevant to preterm babies. And then talk a bit about lactoferrin and why we chose to study it, the large trial that we did, and then touch on some of the mechanistic work that we're still kind of working through at the minute. And one of the kind of questions I want to pose is how difficult can it be to do a high-quality nutritional study? For us as clinicians, the theory is amazing over the last few days. But what we want to know is how much of this is going to change what we do when we go back to the NICU. And this is the feeling of pressure and anxiety that I've been feeling since I've been here. I came fairly kind of confident that I knew what I was talking about, but as I listened to several decades or centuries of research expertise, I've started to get increasingly worried, but was comforted by going to the Olympic Stadium the other day, where Coubertin said, just look ahead, speak frankly, and act boldly. So that's what I'm going to try and do for the next 45 minutes or so. And here's my career CV. I've not won any awards, I haven't got any medals, I haven't got any honours in anything, but I have looked after about 2,000 babies less than a kilo. So that's my credential. So here we are. This is where Newcastle is in the north of England. We have 3 NICUs in a big population area of about 3.5 million. We're the largest unit. We accept all the babies in for complex surgery. All the fetal medicine cases and diaphragmatic hernias are born with us. So we have a very kind of complex mix of babies. It's a beautiful city built on the banks of the River Tyne with a lovely opera house. And it would be impossible for me to come here and give a talk without discussing Brexit. We do agree we would like to move from where we are, but our proposal is that we just move a little bit further south, where the weather is considerably better than it is in Newcastle most of the time. A little bit about the kind of research group. We're a kind of collaborative group of clinicians and scientists that have really kind of just evolved organically over the last sort of 10 years or so. A lot of our work has focused on the microbiome and more recently the metabolome, but we're also doing some viromic work. Chris Stewart, who's done a lot of our work, has developed an enteroid model in collaboration with Janet, who's in the audience here, where we retrieve some tissue at surgery. So babies who go for neck surgery, we get permission to take a little bit of the resected tissue and grow the organoids from stem cells in the lab. And that hopefully will provide us with a very appropriate model for testing some of these interventions in the coming years. So say a lot of our work is really focused on neck, but we do have some long-term follow-up studies looking at the consequences of early life growth on later metabolic outcomes, insulin resistance, and these things. And importantly, we have the Great North Neonatal Biobank, of which Janet is the director. And in this, we have an amazing number of samples from probably over 1,000 babies now. Most of these are stool and urine samples, but we've already set up a system to retrieve salvaged blood. So when you take a blood sample and you send it to the lab and there's a little bit of EDTA or serum left over, the lab kindly put it in a freezer for us so we can go back, because of course looking after 500-gram babies, it's simply not possible to go around taking blood from these babies whenever you want. And in addition to all those samples, we also have some samples of breast milk and tracheal secretions, so that's really what we're trying to do. We've also been involved in setting up some pretty big studies over the last few years. The feeding trial, this was 2,800 babies. Hopefully that will be available in the next few weeks. It's coming out in the New England Journal. The lactoferrin trial ELFIN that we'll talk about, our mechanistic study, MAGPIE, which was nested within the ELFIN trial. But we've also been involved in the trial of exclusive human milk diet funded by Prolacta. So just to kind of set the scene for those of you who are not neonatologists, just to kind of give you the perspective of how difficult it is to feed these babies and just, to show you what their nutrient requirements are. Anybody know who this guy is here? Any Colombians in the audience? This is Egan Bernal, who won the Tour de France this year. These guys cycle up and down mountains for 6 or 7 hours a day, and then they do it the next day and the next day, and they do this for 3 weeks at a time. And during this period, they consume somewhere in the order of 7,000 kilocalories a day, yet they still only weigh about 60 or 70 kilos. And just imagine how wonderful it would be to consume 7,000 calories a day of all this wonderful food, but still be as thin as that. So that's what he needs in terms of energy expenditure to cycle up and down mountains. Compared to that, our babies on the neonatal unit probably require an energy intake of around about 120 to 130 kilocalories a day. Therefore, our babies require 20 to 30% more energy than the most energy-consuming activity known to man. Quite incredible, really. And out of all of that energy expenditure in early life, about 2/3 of that is consumed by the brain. So quite easy to understand if we don't meet macronutrient intakes, why babies may turn out with longer-term problems. So that's energy. And then just to show you protein, so a 24, 25-weeker probably requires somewhere around about 4 grams a kilo a day. To show you what that would mean for you guys or for me, it would require eating about 15 beef burgers a day. I mean, can you imagine how appalling that would be? But that just shows you how incredibly high nutrient requirements are in preterm babies, and therefore the challenges that we face, both in terms of giving them enough parenteral nutrition, but also enteral nutrition, and how we do all that without them getting NEC, necrotising enterocolitis, or sepsis. And, of course, the most important outcome for us is long-term cognitive outcome. The worst outcome for a preterm baby is impaired neurodevelopmental function. And humans, as you know, are quite different from most other primates. Most primates have the rapid period of brain growth prior to term, so they're born relatively mature. Whereas for humans, we know that the rapid period of brain growth happens in the 3rd trimester and the first few months after term date. So that between about 24 weeks and 2 years of age, we acquire about 90% of our final adult brain volume. And when we're thinking about preterm babies, as I say, developmental, motor, cognitive outcome are clearly very important. But as I say, we're also interested in these aspects of metabolism in later life. And there are lots of kind of challenges we face, but here are the four of the kind of very key morbidities that are probably going to be affected by nutritional intakes in some levels, so retinopathy of prematurity, lung disease, but particularly necrotising enterocolitis and sepsis, which is what we're focusing on in this talk today. Again, just to show you how far we've come, this is data from our unit over the last sort of 15, 20 years or so. And what you can see is that, of course, there's a lot of kind of bobbling around because we didn't have so many 23-week babies 20 years ago. But what you see is that at every gestational age over the last 15 years, we've got better. So survival at 23, 24, 25, 26, it's getting better. And just take a note here that in our unit at 23 weeks, our survival, and this means surviving to going home and being alive at 12 months of age, is now in the order of 60 or 70%. That's incredible compared to what we were maybe 20 years ago. And we could spend a lot of time talking about why we've got better. We actually haven't had any new medicines. We've got the same surfactant, the same antibiotics, the same steroids, yet we're doing better. And one of the key factors is an ability to work together, both the nursing team but also the physicians, and this shared combined experience and knowledge is really what's made the difference here rather than a single intervention. But when you think about these 20 or 30% of 23-weekers who don't survive, or the 24-weekers, we now know that after the first few days of life, the most common reason for a premature baby to die is due to neck and sepsis. And both of these key diseases are clearly closely related to our nutritional and feeding practices. So over the years, what we've seen is that better nutrition undoubtedly has led to improved survival. And this is a problem of global relevance because, as you know, around about 20 million babies are born either low birth weight or premature every year. So this is relevant to every country and every continent. But as survival improves, we're faced with the challenges of looking after increasingly complex babies who may have had neck and had surgery and now have short gut. So this is a circular relationship. As we get better in nutrition, we need to learn and know more to look after the complexity that we face. We've heard a lot about breast milk, and as I say, I'm not talking about that specifically today, but clearly breast milk is the most cost-effective intervention in the whole of neonatology. However much we think probiotics, or we don't think probiotics work, breast milk from the mother will always be better than probiotics. There's a lot of evidence that it improves maturation of the brain, it improves maturation of the immune system, but most importantly, it results in better cognitive and metabolic outcomes over the entire life course. So we're not questioning the benefit of breast milk, clearly, as we've heard in lots of talks. The question really for us is, what can we learn from breast milk that may allow us to improve nutrition in these babies? And this is my kind of simplified scheme of nutrition that I've been kind of trying to sort of think and develop, I guess, over the last few years. Because for me, nutrition is more than nutrients. People get very bogged down in neonatal units about grams of protein or micrograms of this and that. But really, the nutrients are just one part of what I consider the nutritional package. So the nutrients are clearly your macronutrients, your micronutrients, etc. But then, as we've heard in some great talks today and yesterday, the role of the functional components, the HMOs, the growth factors, the enzymes, but then also the interactions with the microbial environment and the gut microbiota that we're very interested in. And then I would have the fourth component as the kind of technical socio-behavioral aspects of feeding. Direct breastfeeding, the impact of tubes, sensory experiences, taste, and all these things. So these are the four components, I think, that we need to consider when we're talking about nutritional interventions. This is a picture of Harrison, who is a 23-week surviving baby. Very sadly, his brother died at a few weeks of age. I'm still in contact with his family and his mum. He's now 8 years old. He's an amazing little boy. As you can see here, mum was doing the right thing. Here he is only weighing about 1.5 kilos, and still he's actually trying to breastfeed before discharge. So Harrison's mum was completely committed to breastfeeding. She hated expressing. She said it made her feel like a cow, having to go off to that room 8 times a day and express. She didn't enjoy any of it. But she said she did it because she was convinced that it was best for her baby. So it's a very important message for us in terms of how we support and encourage mums. But I'll be honest that when I started doing nutrition research maybe 25 years ago now, and I was doing studies of different amounts of protein, I kind of just thought about formula as being something that was effectively fluid and nutrients. And you tip it into this long tube, which is the gut. I can see everybody in the front row looking disappointed that I had no idea of all the importance of mucosal immunology. But this is what I thought. I think you just tip it in there, and at the end you produce this waste product. And effectively that's sort of nutrition, isn't it? Just work out how much you're going to tip in and how much you're going to absorb. And of course, what we've all learned over the last 10 or 20 years is that it's much more complex than that. There are all these relationships with metabolites, but also interactions with the immune system, the endocrine system, and your microbes. And so we've had to shift our thinking from thinking of breast milk as a food to being a biochemical fluid that is allowing the mother in some way to perhaps signal to her baby. Let's put this quick slide in here just to remind all of us that although we think about the protein in breast milk as being the kind of casein, the whey fraction, there are lots of different casein components, but proteomic studies have now shown that even breast milk has peptides, that there's some digestion of protein in the gut. So we have proteases there, but then, of course, we have to have anti-proteases to stop breaking it all down. And I think this just reminds us that when we're talking about protein and the peptide fractions, to understand the complexity of protein digestion and really how little we know in preterm infants. And again, this is my kind of simplified schema. This is, protein digestion 101 for medical students or for a lot of us as neonatologists, because we don't get taught this very well. But preterm babies are very different. They tend not to have the low levels of acidity that you have in adults. And so although there are lots of proteases expressed on the brush border, and there are lots of peptidases in the bacteria, and then when you finally get to the bottom end, there are anaerobes that are going to ferment the bacteria. We know virtually nothing about this in preterm babies. So that when we come back to talking about protein and how to lactoferrin and how lactoferrin may be cleaved into lactoferricin, we really don't know what's happening in preterm babies very well. And so although we start off with these complex protein structures, bit by bit as you go down the tract, the protein is denatured and switched into to peptides and amino acids. But as I say, we don't know in preterm babies very much about this. So as I say, we understand there are different kind of protein fractions, caseins and wheys, and the whey compartment particularly contains lots of proteins, alpha-lactalbumin and lactoferrin. And we need to sort of stand back and be impressed by the complexity of human milk and how this whole protein whey-dominated milk, particularly in colostrum, provides this ideal balance of amino acids that optimize brain and metabolic outcomes. So these hydrolyzed proteins and peptides do occur naturally, but as I say, these key steps we know very little about, but we suspect that gut microbiota are going to be very important. And I don't need to tell all of you here in the audience how complex the gut microbiota are, the fact that we should really think of ourselves as a superorganism with all these bacteria being involved in lots of different metabolic and immune processes. But we need to understand that human development would be impossible without these microbes. So if we didn't have these microbes, we wouldn't be able to digest foods in the way we do. And as many of you know, these weigh 1.5 kilos if you add them all up. So again, we can all take some comfort that all that extra food we've eaten over the last 3 days, when you weigh yourself and you get home tomorrow, you can subtract 1.5 kilos because it's not really you. And we're here because we know that the gut is the inner tube of life, and that, as I say, is the focus of our research. There was a nice paper that came out a couple of years ago talking about human evolution in the context of a microbial world, as if this is some sort of struggle, like a kind of tug of war, where on the one hand we have to learn to manage the beneficial microbes, but then exclude the pathogens and the harmful microbes. And if you think about that, that kind of challenge is very acute in a preterm baby, where the baby has to work out which bacteria and which other antigens you should become tolerant to and which ones you should elicit an inflammatory response. And if you think about a 24-week baby who is supposed to be in utero swallowing amniotic fluid, how on earth does the preterm baby know that the bifidos are going to be the good guys and the E. coli are going to be the bad guys? He's had no memory, no teaching to do this. So very easy to understand why gut microbiome development in preterm babies is going to be so complex. And you will know that the pattern of bacteria change over the life course. So what is kind of optimal for newborn babies is very different to when we're older. Major impacts, as we heard from Sammy talking about when you reduce breastfeeding and how that interacts with your microbiome. So we've learned quite a lot about how this changes over the life course. But we really don't know what should be healthy in a preterm baby. And if you kind of extrapolated this graph backwards, what on earth should these patterns look like? Because we have no model of a healthy preterm baby. By definition, every preterm baby is unhealthy. They simply shouldn't be here. And nature didn't evolve to look after them. So trying to work out what microbial community structure is optimal in a preterm baby is incredibly difficult. So we've heard a lot about the interaction with mucosal immune system, between your bacteria, the dendritic cells that reach out and sample these cells and prime and impact with your T regulatory cells, and then how this impacts with the systemic immune system. And when I come to talking about lactoferrin and we start putting some extra lactoferrin in the gut, just thinking about all the different ways in which that might act. So it may act as a food for certain bacteria. There's some data showing it may be probiotic for certain bifidos. It may prevent the growth of other bacteria, particularly those that rely on iron as a source because it binds iron. But there's also data to show that it interacts with the gut epithelium, interacts with T-regulatory cell development. So lots of different mechanisms by which lactoferrin might be working in our babies. And then when I come on to talking about the clinical trials and thinking about sepsis, and in a lot of our babies, we suspect that the coag-negative staph infections that may be associated with catheters or on the skin, this lactoferrin has somehow got to do something getting through all this system if we really believe that lactoferrin is going to stop skin infection in our babies. So this is my simplified schema for clinicians, just to try and sort of understand that we're talking about gut microbial communities. So these are communities of bacteria, and that there's this continual interaction with nutrients in the gut and all the metabolites that they're producing. And then there are lots and lots of steps before you then get to the stool. So whilst we do a lot of our work by sampling stool collected from a diaper, really all this interaction is happening way up higher in the gut. And we simply don't have methods for us to explore what's happening to the microbiome or the metabolome, particularly in the terminal ileum where most of the neck disease seems to occur. So by the time these bacteria or metabolites have got down here, they're likely to be very different. That doesn't mean that we can't use this, but we have to appreciate how restricted we are in studying where we are. So say, welcome to a far, far away place. This bit here is, in microbial terms, 1 million miles from what's happening in the ileum. So we know that there are lots of different dietary components that are likely to impact on gut microbial ecology, so particularly the HMOs, but probably just every nutrient that you study will have some impact on gut microbial communities. This is very different between breastfed and bottle-fed babies. But then in turn, all these microbes are producing other key compounds. We've heard a lot about short-chain fatty acids and how these may prime the immune system. We know that some bacteria produce amino acids that we then may absorb. Lots of bile acid synthesis is associated with gut microbes, production of choline and some key vitamins dependent on microbes. But also a massive array of anti- and pro-inflammatory mediators and signaling molecules. So this is unbelievably complex, and no way a humble clinician like me is going to unravel what is happening, particularly because we can't get to the bits that we need to get to. And we haven't talked a lot about it in the symposium this time round, but we're all starting to understand the importance of the gut-brain axis, this kind of bidirectional biochemical signaling pathway, lots of different sort of routes that connect the gut to the brain. And as I say, when we're thinking about maybe lactoferrin or some of these compounds, thinking about the peptides that may themselves have activity in the brain, I'm a little bit skeptical about how relevant this is in preterm babies, but clearly there's been a lot of work in adults linking bowel pattern, bowel habit to certain diseases. For example, people who develop Parkinson's disease turn out to have a history of constipation that was present years before they developed the symptoms of Parkinson's. So there's some really fascinating relationships to explore. And it's possibly true that in the preterm baby, some aspects of motility may be part of this pathway. But as I say, we really don't have much work on this at the minute. So this is the normal kind of process in healthy term babies. Birth seeds your microbes and genes, essential for all these components. You're supposed to come out covered in maternal secretions. So I like this phrase, birth seeds, breast milk feeds. That's what nature intended. You're supposed to do all these things. And then you're supposed to go home and get some extra seeding from the environment around you. But when you contrast that to a preterm baby, nothing about a preterm baby is anything like what it is for a term baby. Often born by C-section, they're put in clean plastic incubators. We wash and scrub and alcohol gel our hands. Just imagine what bacteria can survive that whole process to colonize the baby. Then we stick needles through their skin. We put bits of plastic that can all be colonized. We feed them with very abnormal regimes. Even the breast milk isn't produced in a normal way. It comes into contact with plastic and is stored before it hits the baby. Then if you're in a naughty unit, you give all these horrible H2 blockers, and then they get lots of antibiotics. So it comes as no surprise that the preterm microbiome is completely different to a term baby. So we've had this sort of diagram earlier before that there is this continual interaction between these three components, between the microbes, the nutrients, and the immune system. And what's optimal at the beginning of your life course is different from what's optimal at the end. And we can think of NEC in a very simplistic fashion as being a breakdown between these three components. So some it may be more immune-driven, some it might be more microbial, some it might be more nutrient. But somehow there's a breakdown between these different elements. And we talked a lot about evolution, and Olaf gave a great talk. I mean, it is absolutely fascinating to think, how breast milk evolved. And everyone says, and this Russian Dobzhansky, nothing in biology makes any sense except in the context of evolution. But what about preterm babies? Because preterm babies have no pressure on evolutionary effects. And we heard from Joe that, we see these changes before babies get NEC. We move into a more kind of dysbiotic-looking pattern, although we're not really quite sure how to define the dysbiotic pattern. But what we don't know is, are the microbes causing the inflammation, or are the microbes just a consequence of the inflammation? Is it nutrients driving the inflammation, which are driving the changes in microbiota? So we really can't unpick this, but we do know that there's a signal there in the microbiota to be had. And NEC, as I say, is the most important thing. And that's because NEC in childhood kills more children than all of childhood leukemia and lymphoma put together. So for a lot of people, this is just seen, this is just a problem that happens in preterm babies on a NICU. We don't need to invest time in it. But you're more likely as a child to have been a preterm baby who dies from NEC than all of these complex oncological problems. And we think about the major advances, thank God, in the treatment and success of these things. But when you think about NEC, really, our treatment hasn't changed in the last 20 years. We're no better at predicting it. We might be a little bit better at preventing it in some babies. But we're looking after more babies who survive. We're caring for increasingly vulnerable babies. Last year, we have our first surviving baby who weighed only about 350 grams. I'm not suggesting that's going to be a common occurrence, but somehow, almost magically, we managed to feed that baby without him getting NEC, and he's now a year old and quite healthy. So this is increasingly kind of complex for us. We're looking after lots of babies after surgery. And I'm going to be the third person to show an iceberg in the talk today, which is thinking that NEC is really just, for me, the tip of the iceberg of gut health. And lurking below it are all these other factors that relate to gut health and outcomes, duration of PN and antibiotics and cognition. So I like to think of a spectrum of gut health rather than a discrete yes, no, did he have NEC? I think if we just go for a yes, no approach, we kind of lose some of the subtlety in what we're talking about. And of course, it's possible that lactoferrin affects all these outcomes. So we use this horrible kind of histological term, necrotising enterocolitis, really for a clinical syndrome. NEC is not a single disease. It's the end result of lots of different pathologies, lots of different kind of causative factors. We never understand the etiology in an individual case, and treatment is specific and crude. So here, this is a baby with black necrotic gut. He's definitely got NEC. Here's a completely healthy baby. At what point along that line do we choose to say this baby now has NEC? And if we can't be sure about where you are on that line, how on earth do we do studies to unpick it? So those of you who are clinicians in the audience, we can have great fun over lunch, arguing over which of these x-rays conclusively shows the baby has NEC, which one doesn't. If I told you that you had 1.4 mils of this coloured aspirate, would that change your opinion? Or 0.5 of that, or really dark 0.5, but we love looking at these things on neonatal units. And then I'll show you a few bellies. Some are a bit distended. Some might have a little bit of umbilical flare. We put all that together, but this is how we make our diagnosis on a neonatal unit. It's really pretty crude, and we're obviously losing a lot of the signaling in all of this. And things are very different between units, so your approach to how readily you will stop feeds or start antibiotics or request an X-ray will change or impact on your instance. We know that there's variation in the rates of babies you get sent for surgery. So if your diagnosis is surgical neck, there's a twofold difference between units and countries. How good is your histology? Do you really check this out? Are you absolutely sure that that was a perforation? Perhaps it was an isolated perforation where you get a secondary peritonitis and you miss it for 24 hours and then it looks like neck. How do we work out whether this was a perforation or neck to start off with? And if you don't review, or you don't have high post-mortem rates, or you don't independently review these things, how can you be sure really what we're talking about? So this is a massive challenge that we face. So we certainly need biomarkers. So what have we been up to in Newcastle? As I say, we've been working on this, particularly Chris Stewart has led a lot of these studies with Janet and myself over the last few years. I could show you lots of pretty pictures. I just want to sort of start off here because we found 12 twins and a triplet, and we looked at the stool microbiome. And what we were impressed by was how closely siblings clustered with each other. Maybe that's not surprising, but these babies are in different incubators. They're being touched by different nurses. Some are having antibiotics, some are not. But they're actually all quite similar. So presumably the breast milk exposures and maybe some genetic aspects really are quite important of this. So I'm going to come back to twins in a little bit. We were also able to look at the microbiome and show how closely the breast milk microbiome maps to the stool microbiome in some of these babies. So again, as we've said, we know that breast milk is a key driver of this. But what we've also been able to do is say with Janet's sort of painstaking collection of stool samples, if you want a stool sample from a preterm baby, go speak to Janet. She's literally got hundreds of thousands of them. But we could really look at the development over time. So here are twins where one twin gets neck and the other doesn't. So the red one doesn't get neck and the blue one does, and this is Shannon diversity. And you can see that maybe just a few days prior to neck, there is lower diversity as you might expect in the baby with neck. But if you go back a little bit earlier, there really wasn't any difference. So how on earth could we have predicted which of those was going to get neck? And we're then also able to look at the differences in taxa and show how things changed in the baby prior to neck, where you go, oh, look, increase in Klebsiella, that's not surprising. But then why didn't he get it here where he has more E. coli or something else? So it's just so difficult in individuals to predict what's going on. But also look here, these are where we put the antibiotics into the baby. Pen and Gent, and then Fluclox and Mox-Gent, and these things. These antibiotics don't have the big impact on the microbiome that you might expect. Clearly they're important, but we're not able to really manipulate the microbiome as much as we would think. So this was a great paper from Chris looking at 40 babies with lots of longitudinal samples, and I'm just going to show you how chaotic the microbiome is. So at the bottom, these dashed lines, these are the core OTUs. These are either Klebsiellas or E. colis or enterococcuses, and the top of the satellite ones, these are the ones that are in much lower densities, things like bifidos. So here's one baby, just an example, just to kind of get your eye in. Over days 1, 2, 3, 4, 5, 6, 7, 8 of life. So he has Staph aureus, it disappears, then it comes back again. E. coli, everything in the baby at this point in time is E. coli, goes away again. Bifido, up, down. So completely random in this baby over a period of a few days. And then this is what it looks like in 40 babies. So just stand back and look at that and just look how different and how chaotic it is. How on earth are we going to unravel what's going on here? And baby 234 is this one here. 100% of everything in that baby's gut for 5 days apparently was Klebsiella. Like, I wouldn't like that to be my gut. But he was absolutely fine. So I think the key message from all this is that this is completely chaotic. And every baby is unique, and working out the disease one is impossible. So here's a heat map where Chris applied some clever statistics to break them down into different community types. And the computer model recommended that we would have 6 different community types. 1 is dominated by Klebsiella, 2 by Klebsiella enterocolitis, etc., etc. Note that 6 is the only one with a substantial number of bifidos. And so each of these, I think there's about 800 samples here. Anyhow, so we have 6 community types. So if you hold those 6 community types in your head, this is what we did next. So we replaced each of those community types by a little dotted color for the next graph. And then this is what it looks like. These are all the control babies, these are the NEC cases, and these are the sepsis cases. And you can see that each day we have a sample, we put them into one of those 6 different types. And what you see is that early on, maybe not surprisingly, there's a lot of staph dominating the infant gut. And then as time goes on, you start to see more bifido and more diverse appearances. But what we were able to see was that in the babies who got NEC, they had many, many more transitions. They were moving between their community types much more frequently than the controls who appeared to be a bit more stable. So this was significant, of course, a whole lot of statistics there, so you can take it with a pinch of salt. But we do believe there's something about the chaos that's going on in those babies' gut that might be associated with NEC, rather than just specific types of taxa. I don't know if they're paying attention. There's an animation here, if you can click on it. I haven't got a clicker. That shows a baby moving between the different community types. Can someone ask oh yeah, there should be a bar at the bottom where you can actually move the end. So yeah, so this is a baby moving around before he gets NEC in red, and then coming in green is a baby who's healthy. And what you see in the baby with NEC is that he's moving all over the place before he gets NEC. Whereas the baby who stays healthy kind of moves to one kind of community type and then just kind of keeps around there. So that's a bit stylized because we obviously chose 2 examples that make the point. But I think there is something here about the kind of chaos that is associated with NEC. So say NEC's a major challenge. We've learned a lot, but we still don't know how to prevent it. Mechanistic understanding is really very limited. The animal models, as Joe was pointing out yesterday, I think are very, very limited for our babies. And of course, we have lots of ethical and practical challenges. But the fear of NEC dominates decision-making. And as I say, we should think of NEC as being at one end of the spectrum. So the question is, does lactoferrin reduce it? All of you will know about lactoferrin, this important antimicrobial whey protein present in colostrum, breast milk, tears, saliva. Then the presence of acid is converted into lactoferricin, which again has antimicrobial, antiviral activity. Does this happen in a preterm baby in the same way? We don't know. We know that concentrations are particularly high in colostrum, and we know that all mammals express lactoferrin. And again, reading lots of the papers from Professor Oftedal and his group, understanding how these proteins existed before we were even mammals, that how you'll find lactoferrin in worms is just amazing, how this protein has persisted. And there are hundreds, literally thousands of papers showing different activities and impacts of lactoferrin, which I'm not going to go through, but you can just take it that studies have shown impacts on microbes, viruses, fungi, lymphoid development. Lots of reasons this may be important in preterm babies. Lots of our babies get biofilms on their catheters, and lactoferrin may prevent this, prevents adhesion, disrupts virulence proteins. So lots and lots of theory that I'm not really going to get bogged down in today. So say we agree lactoferrin from your own mother is probably the best source, but these babies get really very little amounts of milk in the first few days. And a lot of mums don't provide sufficient milk. Human recombinant lactoferrin is available. There are some challenges with the studies, but it's likely to be very expensive. So what about using bovine lactoferrin? Widely available, probably pretty cheap. Similar in structure, but is it similar enough? So what do the Cochrane meta-analysis show on whether lactoferrin would be a good thing for preterm babies? So this came out a couple of years ago. These are all the trials in preterm babies. looking at the impact of lactoferrin on sepsis and NEC. And what you see is that in 6 trials, you have a significant reduction in sepsis, and in 4 trials, a significant reduction in NEC. So this looked like a very promising intervention for us to try. Just note also that this is in the wrong place here. So in the Sherman study, there was also a significant impact. So this is a sort of an error, printed or typo or something we need to correct. Anyhow, a lot of data there suggesting this could be a great intervention for our babies. So one of the challenges were all the trials were underpowered. I think there are issues about blinding and masking. And as we'll come on to, the source and quality of lactoferrin may differ between these trials. But we clearly felt we were justified in trying to put together a large, well-powered trial. If lactoferrin, that it was likely to be very safe, likely to be cheap, really does reduce NEC or sepsis, even if it only reduces it by a small amount, what a wonderful intervention we could have. This is our unit. This is what a neonatal unit looks like. It is unbelievably complex. A lot of technology, a lot of equipment, very sick babies, very stressed parents. And trying to do clinical trials in this setting is incredibly challenging. And we're still learning. We've been doing these trials for 20 years now. But running trials in this environment is, as I say, incredibly challenging. So how do you do a good quality trial? Well, you need to make sure that you're asking a question that your users, your parents, the public think is important. You've got to choose a great intervention. You've got to work out how much of the intervention to give and for how long and in which population. Then you've got to be really sure that you choose a robust outcome that's really meaningful. We wouldn't get funding to do a big trial like this without powering it at 90%. You can't do it at 80%. The funders just would tell you to go away at that price. And then when we went to get funding, we're competitive with the adults, doing huge trials of cardiovascular disease. So where did our trial start? Well, about 15 years ago, we put together a multidisciplinary group of neonatologists and dietitians. We formed in 2003. We meet 2 or 3 times a year. We're not aligned to any organization. We're completely independent. And this is our meeting in 2009. And I say, we're just clinicians. None of us had any academic time in our contracts. Here, the 6 of us here, we were the founder members from 2003. And then everybody else starts to come along when they see it's like a good idea. And Bill, who's sitting in the back here, had this great idea we should do a trial of something called lactoferrin. I had this amazing idea that we should do a trial of early or late feeding in neonates. And I was so wedded to that I really just thought it was important I did a trial that was called ELFIN. Bill came along and decided that he would actually take the lactoferrin on, and he stole the acronym from me to being Enteral Lactoferrin in Neonates. So not only did he take my acronym, he decided to do the trial. John here decided to do this trial but call it something different. And I was left to literally pick up the shit after they'd chosen what they wanted to do. So the question was, does supplemental bovine lactoferrin in preterm infants less than 32 weeks at that dose compared to placebo, which is sucrose, reduce sepsis? And we had a very robust case definition. This is the timeline. This is just to show you scientists how hard it is to get one decent paper out. 2009, sounds like a good idea. Let's put an application in. The SIF guys went, yeah, this is a good idea as well. They put their application a bit later. We were working on both of these together. Then you're going to get funding. Then the first supplier of lactoferrin pulled out. Then we had trouble working out what placebo. Then we had to get approvals. Look, we're already into 2012 here. So obtaining the product and designing the trial was really complex. You'll see Janet here sitting there laughing at our ability to try and work out how to blind it, because some of these things here are lactoferrin and some of these things here are sugar. So I thought we'd play a little game now to see whether you think we got the blinding right. Here you can see us playing around with lots of stuff on a desk in our seminar room. So number 1 here, who thinks that's lactoferrin? Who thinks that's sugar? Hands up for lactoferrin.
It depends on the degree of saturation with iron.
Yeah, that's cheating. Okay, so this one was lactoferrin. This one here, lactoferrin or sugar? Recombinant lactoferrin. 1-1. This one here, it's got to be sugar, hasn't it? This one here. This one here. Lactoferrin. This one here.
I'm unsaturated lactoferrin.
No. Cocaine. And when people came in and saw us at this table with all these different powders and started taking photos, we were worried that whether Bill was going to turn into our Colombian friend here. Okay, there was no food dye licensed for use in preterm babies. So we went with using brown sugar in the end, because at least it was different from sort of white sugar. Lactoferrin is nothing like maltodextrin, which is a powder. So we tried really hard. We had a lot of quality control and assurance over our products, a lot of complexities with European law. We used a product from New Zealand. We had to import it in. The first time a big bag of white powder arrived, it was sent back because they weren't sure what it was. We obviously need to make sure it was lactoferrin, so we know it was definitely lactoferrin. But as we can discuss, like, we only know it's lactoferrin. We don't know about the bioactivity. We knew a lot about iron binding, so of course very important that we had a product that was not too iron saturated. A lot of quality control about how long that would be stable for, 12 months, 18 months. We bought it from New Zealand because they've not had any of these nasty viruses. We had to make sure it wasn't contaminated with metals. You're giving it to vulnerable babies. Then when we finally got the stuff, and of course if you shake to dissolve it, you realize it froths because it's a protein, whereas sugar doesn't froth. So then we realized that if we put a little bit of water with the lactoferrin plus 1 ml of the mother's milk, and then you shake it, both the placebo and the lactoferrin froth. You then got to set for 30 minutes whilst the bubble disappears. But the lactoferrin was still a little bit different coloured, not surprisingly. But we had these purple syringes that provide a little bit of masking as well. So this was our actual lactoferrin. We put it in these pots. We then put a stopper on the pot so that nobody could look inside and try and work out what was what. And then you connect a syringe to this popper. You make this up into batches, 1 each day of life, and you provide them to unit. Straightforward, problem solved, until you realize that every hospital, and we had 37 recruiting hospitals, has a different type of purple syringe, and they don't all connect to each other, and worse still, they don't all connect to the same NG tubes. So then somehow we had to sort of work around that problem. We then get inspected by the directorate to make sure that we're doing everything properly. Next problem was we were allowed to store it at room temperature, which is officially less than 25 centigrade. But if any of you have been on a neonatal unit, you regularly get over 25. So we had to monitor temperatures and make sure that we could keep it and check it. And all the time, the clock's ticking. So here's where we published it. Here's where we started. 10 years of our life. So can you imagine the complexity in providing this to 134 different sites, developing algorithms, randomization programs? We were also setting up the feeding trial at the same time. So this was all going on at the same time. That's only just published now. And then almost 5,000 babies between those two trials. There was a little bit of crossover, about 200 babies in both. And then we try and set up to sample lots of stool and urine. So I didn't look anything like this 10 years ago. I had very blonde hair, no wrinkles. This has been a little bit wearing for all of us. So we recruited 2,200 babies. We have the primary outcome in 99.5%, very low incidence of adverse effects. But late onset sepsis was no different in controls to lactoferrin. And this is the chief investigator when he finds that result. I mean, we were absolutely gutted. We set out because we thought there would be something there. Of course, we had equipoise. That's why we did the trial. But we didn't set out to prove this doesn't work. Why would you do that? So we were disappointed. And everyone said, well, surely it made something better. Maybe some of these other, retinopathy or BPD. Nope, doesn't make any of that better. People go, oh yeah, but do subgroups. It must work in the small babies or something like that. So this was pre-specified. Does it work in small babies or just those on breast milk or just those on formula milk? Nope. So we all sat around going, what on earth did we do wrong? And this was the principal investigator meeting once we all had the results. We spent a lot of time crying, working out what did we do wrong. But we told ourselves, look, maybe it's just true that it doesn't prevent sepsis. At least the lactoferrin we used in the trial we did doesn't work. I'm not saying, of course, the lactoferrin works. It just doesn't work to answer this question. So why do we do it? Well, Manzoni had a high-quality trial, 470 babies, a 3-arm trial. These are always complex. They always give you concerns about power, but basically lactoferrin, and a probiotic or placebo. They showed really quite a significant result, 5.9%, only 4.6% sepsis in the lactoferrin and LGG group compared to 17% in placebo. That's a massive effect. Reduction in sepsis-associated mortality. No babies died of sepsis who got lactoferrin compared to 4.8%. And then they did this add-on trial that has raised a few issues. Sherman did a trial published a couple of years back using tau lactoferrin, 120 babies, giving every 12 hours. They see less sepsis, 17% versus 33%. They also did some stool microbiota profiling. I'm not going to go into that detail, but it did show differences, lower Enterobacters and Klebsiellas. Sounds like a nice outcome. But this was only in 23 out of the 120 babies, so really just a really tiny number of babies. So why might we have seen things different? Obviously, difference in methodology, lactoferrin preparation. The babies are different. We gave more per dose than in the Manzoni trial. Interestingly, 5% of the infections in Manzoni were fungal ones, whereas we very rarely see fungal infections in the UK now because people use prophylaxis. A lot of our neck and sepsis occurs in the first 10 days or so. So really, how much lactoferrin will they have received in that time? What about if a baby is put nil by mouth? This is an intention to treat analysis. And if you really just have 5 days of an enteral protein, do we really think that that's going to exert an effect at a peripheral site like an IV, stopping that baby getting sepsis? So lots of other factors. Maybe lactoferrin needs osteopontin or some other cofactors for it really to work. Maybe the level of iron binding was a bit too high. Maybe there's some problems with transport and storage. We were convinced that we have very good high quality control. We have to pasteurize it. I've had one person say, you shouldn't have pasteurized it, but seriously, would you risk giving a bovine protein that wasn't pasteurized? That's not possible for our babies. So, of course, bioactivity is going to be key, and we simply don't have a method for knowing whether the lactoferrin we used really was the optimal product we could have used or not. We were restricted by what we could get hold of. Is it synergistic with probiotics? Well, a third of our babies did receive probiotics. No evidence of synergy. So maybe it's the New Zealand cows that are responsible for our failure to get it into a better journal. Okay, so I'm not really going to talk about this in detail, but just to say we've used the ELFIN trial and the SIF trials, which is our MAGPIE study, mechanisms affecting the gut of preterm infants, where we collect the stool and the urine. We're doing some microbiome metabolomic work, very similar to what Ollie presented earlier on. And in our unit, we've been salvaging blood and doing some immune work as well. So we have 480 babies who are randomized in the lactoferrin trial. With comprehensive data, we know every day what these babies were getting, whether they were getting probiotics or antibiotics and what they were getting. We've got 30,000 samples from these, and we're currently, these are all going through the machines at the minute. So data will be available soon. And of course, we hope to sort of understand what lactoferrin was doing and why it may not have prevented NEC. And we're going to use this kind of twin design that I talked about, because what we found was that at least 100 of our babies in the trial were twins. So if you have twins that are both healthy, that neither one of them get NEC, what we did is we found, of course, that half of those twins will have got the same intervention. They'll both be on lactoferrin or they'll both be on placebo. And half will be discordant. One will have got lactoferrin, one will have got placebo. And to some extent, then we can additionally control for all the other things we've measured. Maybe we're controlling for some genetic factors here or breast milk exposures. In the babies, you get disease. We're obviously looking at how the patterns change before they get disease. And we're using a match that is gestation, postnatal age, and hospital site matched if they haven't got a twin. Metabolomics, this has been a massive learning for all of us who are clinicians. This is really hard to get your head around and understand the new techniques as they come in. But we're doing a mixture of GCMS and LCMS. We're looking at stool, obviously, for products of microbial metabolism, but also looking at urine and plasma. Lots of challenges, but we're working with a great team who've got an extensive library of metabolites. And just to sort of say here, just in case it's true, but this is my prediction, I honestly think that there's going to be some really important signals in urine, and I can see how we could move to using urine as a sort of pattern recognition in our preterm babies, identifying the babies who maybe have some ongoing inflammation or high levels of protein breakdown or something like this. So urine's a great fluid. Stool's very problematic because the babies don't poop very much in the first few days, and if they get sick and have an ileus, they stop pooping completely, whereas the urine keeps coming. So I think urine could be a really important fluid that we need to explore. So these volatile organic compounds that you get on GCMS, we all have our own unique smell print. If you've got kids, you know which one is your kid and which one is somebody else's. This model's been applied to neck. Again, this could be a bedside technology. So here's a healthy twin, and here's a twin with neck, and you see a loss of one of these organic compounds. I'm not sure if they've identified it yet, but it's starting to suggest that you could maybe use this as a bedside technology. And it has already been used to detect rotavirus and C. difficile quite accurately in clinical settings. As I say, the group that we're part of is doing lots of other work, transcriptome and more histology, using stored fixed paraffin embedded tissue from the lab. We can go back to the pathology archives, dig out all the samples with neck, and then do some work with these. We need better biomarkers. I'm not going to go through this, but obviously we're thinking about the stool component, the stool compartment, the urine compartment, and the blood compartment. And we're thinking about what are the different markers that maybe we could use to allow us to better predict. So these may be relevant at different stages of the neck disease in terms of preventing, predicting, but also importantly allowing us to subtype, classify neck in these babies, but also do more mechanistic studies. So hopefully that's going to come over the next few years. So summary lessons, clinical trials in nutrition, incredibly complex and very, very hard work. What works in the lab may not work in the NICU. Nutrients don't function in isolation. Maybe it's artificial just to pick one out and hope it's going to work. Observational microbiome studies are always confounded, sometimes very heavily confounded. At least we've done an RCT. Bovine lactoferrin isn't a human lactoferrin. Of course, we should focus on fresh breast milk, but as we said, we need better assays for bioactivity. But if you really want to know the answer in a clinical setting, you've got to collaborate, do high quality, i.e. large trials with functional outcomes, if we really want to move all of this amazing scientific knowledge to actually improving the health of the babies that we look after. So a massive thanks to everybody who's part of the team, Janet who's in the audience, and Chris who've done a lot of it, Chris Lamb in the lab, Greg Young who looked after all our samples, our funders. Very importantly, all the babies and the parents who trusted us with their babies' lives to do these studies. So as I say, good research is a massive team effort. I'm just presenting on behalf of some amazing collaborators. And I'm going to finish here. We've got a website for our group, neonatalresearch.net. We've got a tab here where we put all our slides. So if you want to go and read these slides or other slides, you can go and get it. And I'm going to finish there. So thank you very much.
Yes, Dr. Mestecki first, and then Dr. Ogren.
First of all, lactoferrin is a glycoprotein. 10% molecular mass is contributed by N-linked carbohydrates, classical ones, and acetylglucosamine, fucose, and so on. But antibacterial activity of lactoferrin is strictly dependent on the presence of iron. Yeah. It can bind to atoms of iron, bivalent iron per molecule. And that's why you show the white versus brown. Yeah. If it's iron saturated, it's brown. And if you add EDTA and remove iron, it will be a white powder. So it depends. my question is, you obviously don't know the saturation with iron because only unsaturated lactoferrin is
I think
antibacterial.
the data is there. The company who provided us with this provided us with the level of iron saturation. I think it was 8% and it was stable.
Is it saturated or not?
Unsaturated, only 8% was saturated is my recollection of the data.
So that have you discovered, have you detected any antibacterial activity? Lots of papers how to do that.
Yeah, we have all the stool microbiome samples, but we haven't got the data to share at the
Best
minute.
paper, Science, John Eldridge, 1980.
Yeah.
Gorgeous paper about degree of saturation and spectrum of bacteria with which it will
So,
interact.
I mean, we completely agree that the actual type of lactoferrin you use is going to be key to it. As I say, we did choose the product carefully. We had to choose a product that we would be allowed to use. We had to use something that was going to be available in sufficient quantities, that passed all the quality control regulations, and that was not heavily iron saturated. But you could argue, or we could argue all day about what would be the optimal formulation of lactoferrin to
If
trial.
you mix it with whatever fluid which you give to the child per orally, if it has iron in it, you will saturate it again and it will lose its activity.
we mixed it with water and then with breast milk, and mum's breast milk has very low levels of iron in it. So like that's the best that we could do. I mean, practically we have to find a way of getting it into the baby. So yeah.
Dr. Ogra.
Thank you so much. It was a wonderful talk. Jerry asked you part of the question, and I hope you are not disappointed. You didn't get any awards so far. But I'm sure you will get with number of patents you can have. But I just want to go back to lactoferrin itself. There are some very early studies with lactoferrin with 6-3 IgA, which I thought Jerry might ask you. In endogenous production in the mucosal tissues, you know. And have you looked at endogenous production in the respiratory tract or in the gut? And this may again be related to the saturation with iron. And that may be quite different, maybe functioning as effectively, maybe more.
Yeah, I mean, of course, we didn't set out to explore the role of endogenously produced lactoferrin. I mean, clearly that's very important.
Why not?
Because I've got to look after a lot of babies as well, and I spend my time intubating, sticking drips in, and running clinical trials. I mean, if our whole research team was focused on lactoferrin, of course, this is where you would go. But as I say, at the end of the day, I'm a clinician, and my interest is in whether some of these ideas about HMOs, lactoferrins, choline, MFGM make a difference in the baby. And as I say, like, the theory is amazing. But at the end of the day, we have to take the step to decide whether you provide a baby at risk of whatever problem, cognitive, infective, whatever. You have to take the step to prove that some of this theory makes a difference. So that's why we chose to do this trial. But I agree, there's lots of other aspects of lactoferrin you could explore in these babies.
Thank you.
Yeah, Dr. Tlaskova.
It is understanding you correctly that in the resected tissue of neck patients, you do enteroids, you get enteroids?
Yes.
Because that's a phenomenal way to get information before you do your clinical trials.
Yeah. I mean, I think one of the challenges in neonatology is that there hasn't been enough basic science in the preterm babies. We still don't really understand the mechanisms leading to neck. So the enteroid system, I don't know how many we've got growing, but 20, 30, something like this are growing. So parents are very happy to support us. The surgeon doesn't resect any more tissue, but he just takes a piece of tissue that's not necrosed, and then Chris takes it off to the lab. So he has these enteroids growing. And as you say, that then provides us with a model, because these cell lines come from premature baby gut, and most often premature baby guts that are at risk of neck, because by definition they had neck. So we have a few controls for spontaneous intestinal perforations. We have a few later controls if a baby has like a stoma closure or something like that. So yeah, we hope that in the years to come these will be useful.
It's very expensive to do the clinical trials, and this gives you a lot of information about what your protocol is going to be.
Yeah, absolutely. We launched in clinical trial because 10 years ago, Manzoni wrote a paper that said this stops babies dying of neck and this stops babies dying of sepsis, and we wanted to crack on and see whether that was true. So if we could do all this in parallel, we would. Sadly, I don't know what it's like in the rest of the world, but getting funding in the UK for this is almost impossible. We're basically full-time clinicians trying to do these trials. So.
Our last question from Dr. Tlaskalova.
It is supposed that very important factor for in complex neck pathogenesis is not well-functioning barrier function. What is known about the effect of lactoferrin in this direction, and are there some clinical approaches to improve this function?
Yeah, it's a great question. The answer is nothing. I mean, there is literally nothing on the mechanisms by which lactoferrin may work in these preterm babies. There are half a dozen small trials, and I know there's a lot of work in toddlers and infants and things like this, but there's nothing in preterm babies, and the physiology of the gut and the nutrient exposures and the microbiome and everything is so completely different in preterm babies that I don't know that we can extrapolate anything that happens in a healthy toddler to a preterm baby.
May
With that, I think
add
we have
one really important information. Lactoferrin is not only a free protein in milk and whatever, it is also within phagocytic cells,
Yeah.
polymorphonucleoleucocytes, macrophages. So if you have inflammation in the tissue, you have plenty of lactoferrin, but it's not because of milk, but it's released by the cells.
Yeah.
With this, I think we have to finish the session. I would like to thank Nick again for a very nice presentation.